In this video, we are going to learn about the smooth muscle in detail. This picture shows the different locations of the smooth muscle. So, the smooth muscle is located in the eyes, especially in the ciliary muscles and iris of the eye, and then in the blood vessels, respiratory tract, gastrointestinal tract, uterus, urinary bladder, etc. And in each of these locations, smooth muscle contraction serves different functions. For example, in the eye, the smooth muscle that is present in the iris will control the size of the pupil, thereby regulating the amount of light that enters the retina. And the smooth muscle that is present in the gastrointestinal tract will regulate the luminal diameter, as well as it helps in mixing of the luminal contents, as well as it causes propulsion of the luminal contents from the mouth towards the anus. So likewise, it serves different functions in each of these locations, and hence smooth muscle is a diverse type of muscle and it is regulated by multiple factors. So the objectives of today's class will be to learn about the types of smooth muscle, structure of smooth muscle, control of smooth muscle contraction, molecular basis of contraction and relaxation, and finally about the properties of smooth muscle. Let us first learn about the different types of smooth muscle. So, there are two types of smooth muscle. One is multi-unit smooth muscle, the other is unitary smooth muscle. Multi-unit smooth muscle consists of discrete muscle fibers, and as you can see, these muscle fibers are not interconnected, and each muscle fiber is separately innervated by a nerve, and each muscle fiber will contract independently of each other. So, that is the characteristic feature of multi-unit smooth muscle. Whereas the unitary smooth muscle, they occur in a sheet, and they are all interconnected by means of gap junctions. So, when one cell is stimulated, all the other cell gets stimulated because of the chemical communication through these gap junctions, and hence it is called as syncytial smooth muscle. And the location of multi-unit smooth muscle is in the ciliary muscles of the eye, iris of the eye, pilorecta muscles, etc. And unitary smooth muscle is located in the hollow viscera, for example, blood vessels, respiratory tract, gastrointestinal tract, and urogenital tract. And since this unitary smooth muscle is present surrounding the hollow viscera, it is otherwise called as visceral smooth muscle. So, there are different names for unitary smooth muscle such as syncytial smooth muscle and visceral smooth muscle. The structure of the smooth muscle. So, this is the structure of a single smooth muscle cell. As you can see, the smooth muscles are very small when compared to that of the skeletal muscle. It's only about 2 to 4 micrometer in diameter in the center, and it tapers towards the end. So, it is spindle shaped cell and with a central nucleus, and these are the dense bodies. So, these dense bodies are analogous to Z lines in skeletal muscle. So, Z lines are absent in smooth muscle, and these dense bodies can be either located in the cytosol or it can be membrane bound. So, the importance of this membrane bound dense bodies are that these dense bodies are linked with the membrane bound dense bodies of the adjacent cells, and as a result, the force of contraction that is developed in one cell can be transmitted to the adjacent cells very easily through these dense bodies. And another important feature is that these dense bodies give rise to or gives attachment to the thin filaments. So, these are the thin filaments. So, this entire structure forms the sarcomere. So, as you can see in this picture, the sarcomere, that is the thin and thick filaments of the sarcomere, are not uniformly arranged. They are not uniformly arranged like that of the skeletal muscle, and hence the striations are absent in smooth muscle, and hence it is called as smooth muscle. So, these are the thin filaments, and in the center this is the thick filament. So, thin filaments consist of actin and tropomyosin. So, remember, troponin is absent. There is no troponin in smooth muscle. So, the thin filaments contain the actin filaments and tropomyosin, and these are the thick filaments that contain the myosin filaments. So, the structure are similar to that of the skeletal muscle, and one important feature is that the content of myosin filaments as a whole is less in smooth muscle when compared to that of the skeletal muscle. And again, the T tubules are absent. So, the T tubules are absent in smooth muscle. Instead, you have this caveolae. Caveolae are shallow invaginations of the sarcolemma. Sarcolemma is the membrane of the muscle cell, and this invagination is known as caveolae, and it is analogous to T tubules that are present in skeletal muscle. And these tubular structures that is present underneath the caveolae are the sarcoplasmic reticulum. So, you will be knowing the function of the sarcoplasmic reticulum, that is, it stores the intracellular calcium. So, as you can see, the sarcoplasmic reticulum is not very well developed when compared to that of the skeletal muscle, and hence the intracellular storage of calcium is less. So, the smooth muscle contraction depends mainly on the calcium that is present in the extracellular fluid. Now, we will see what are the calcium channels that are present in caveolae and the sarcoplasmic reticulum. So, first we are going to see about the channels that will cause increase in the intracellular calcium. So, what are the channels that cause increase in the intracellular calcium? First is the voltage gated calcium channel that is present in the caveolae, which is analogous to dihydropyridine receptor present in the skeletal muscle. And next is the Raaonline receptor and IP3 gated calcium channel. These are present in the sarcoplasmic reticulum. So, voltage gated calcium channel is present in the sarcolemma in the caveolae, and Raaonline receptor and IP3 gated calcium channels are present in the sarcoplasmic reticulum. So, this voltage gated calcium channel will cause the movement of calcium from the extracellular fluid into the sarcoplasm, whereas this Raaonline receptor and IP3 gated channels will cause movement of calcium from the sarcoplasmic reticulum into the sarcoplasm. So, what is this IP3? So, this is a new channel that is present in smooth muscle which is absent in skeletal muscle. So, what is this IP3? It is nothing but inositol triphosphate. So, I will be telling about this channel in detail later. So, for now just remember that this is nothing but inositol triphosphate. And next, coming to the channels that will cause decrease in the intracellular calcium. So, they are calcium ATPase and 3 sodium 1 calcium antiporter. So, these two channels are present in the sarcolemma. And second, Cerca pump. So, you will be knowing about the Cerca pump which is present in the skeletal muscle. What is Cerca pump? It is nothing but sarcoplasmic and endoplasmic reticulum calcium ATPase pump. So, this calcium ATPase pump and the 3 sodium 1 calcium antiporter will result in the movement of calcium from the sarcoplasm into the extracellular fluid, whereas the Cerca pump is present in the sarcoplasmic reticulum, and that will cause the movement of calcium from the sarcoplasm into the sarcoplasmic reticulum.